We've been doing light sheet imaging for just over a year now, but I've been doing clearing and imaging for many years, 30 plus years. Um, so without any ado, here's the outline of what I'm going to talk about. So I'm going to give an overview of Cincinnati Children's and the Research Foundation, as well as our facility. I'm going to talk about the challenges of operating a large facility in a large academic medical center. We have a very diverse user base with things from basic research and model organisms all the way to doing patient-based tissue. Talk about tissue clearing and light sheet imaging, and we are using the Bruker LCS SPIM system. I'm going to show some examples. These would be model organisms down to whole imaging of human tissue from patient samples. Then at the end, I'm going to talk about some image and sample processing to prove our data analysis and segmentation. So, quick summary of our institute and the bioimaging analysis facility. So Cincinnati Children's Hospital Medical Center is an academic medical center affiliated with University of Cincinnati College of Medicine. We are one of the top-ranked children's hospitals in the U.S. and top 3 in research funding for pediatric research. Research being performed is anything from basic research on model organisms and organoids, translational research on organoids and xenotransplants, up to research on patient-derived samples. A brief overview of our facility: we have 11 point scanning confocals, spinning disk, 2 light sheet systems. I'll focus on the LCS here. Wide field stereo microscopes and 9 high-end offline workstations. 5 of those have a Maris full, and we have a number of all the systems have Nikon elements. Yearly usage is for single photon, multiphoton, and light sheet. We do about 27,000 hours a year, 7,000 hours of wide field stereo microscopes, and 35,000 hours of analysis. Again, we have 530 active users from 165 labs at Children's and University of Cincinnati. Notable, we're a Nikon Center of Excellence since 2014, and we have expertise in basically all imaging and analysis modes, other than EM. So this is an important slide. Ignore the tough stuff at the top. Is from another talk. But my team, Amanda Barboza and Mairead Harris, they run the show. They are responsible for all the operations. I do a lot of training and hands-on stuff as well, including developing tissue clearing, but they're the ones that do the work. So my background, again, as Jan said, I got my Ph.D. from the University of Minnesota. I'm a developmental biologist. I've studied axial and germ layer patterning in Xenopus. Xenopus is probably the hardest organism to image in, which does not kill you, makes you stronger. So I founded the core in 2011, 2012, because we didn't have an integrated imaging facility here. I have no disclosures. Disclaimer: I'm not an expert in optical physics, right? I'm a basic biologist. I am a bench researcher. I have been clearing tissue for a long time, and I remember looking at my first cleared Xenopus embryo, and it was just astounding that you could turn this opaque thing transparent. And in this case, it was BAB using beta-gal staining to look as a lineage trace. So I've been doing this for a while. The challenges of running an imaging facility in a large academic medical center is that we have many, many PIs and users with diverse imaging and analysis needs. This could go from live imaging of C. elegans embryos, high mag, high speed to determine Wnt signaling dynamics, cleared patient-derived spheroids, so translational data that we're assessing cell differentiation and different patient mutations, or even imaging patient samples. And on the right is a image from a 2-photon microscope looking at cleared lung tissue from a pulmonary alveolar microlithias patient. This disease, they get basically stones of calcium phosphate within the lungs. And we were able to image the positions of those relative to the airways, the collagen in red, and get some ideas of how these are forming. They form like hailstones, a lamellar structure. So you can see a huge variety. When we added the light sheet, we had this huge variety of researchers and types of tissues, so it's a challenge. So some of the challenges are sample diversity. Investigator experience is variable. There are people working with collaborators that may already have existing clearing protocols that they want to continue to use. The clearing methods, anything from organic to aqueous to hydrogel or combinations of those. Final refractive index differences are also challenging. We have with a immersion lens that are tied to one particular RI, it's tough to image samples, diverse samples that are outside of that range. Labeling methods are also different. And resolution requirements. We are using the LCS, which is great for a large sample overview, but for very high resolution subcellular, it's not the ideal system, but we make this work, and I'll show you how we make this work. So currently on our LCS SPIM system, we have 34 individual users from 24 labs, approximately 180 hours a month on this system, and there are 7 main clearing methods being used. You can see prior to acquiring the LCS, we were imaging with high MAG, high NA immersion objectives, mounting samples on an upright confocal microscope. That's challenging, it's slow, and it's great for moderate to high resolution, but if you're trying to get overviews, it's not very feasible. Now we have the LCS SPIM, we added in late 2023. We have 6 laser lines from 405 up to 785 on this system. You notice the most important thing in this picture is little yellow cables. Those are optical fibers. Data from these systems is amazing. Huge amounts of data. So quick anatomy of the LCS SPIM. We are using a system where the sample is mounted in a cuvette. I think you can see my laser pointer help. The cuvette goes between two objectives to create the sheet, which are shown schematic here. The detection objective is below, which you can see at the bottom of this picture and schematized here. The Z tiling occurred with the stage movement. The X tiling also occurs with the stage movement. The Y tiling is done by objective movement, and the corresponding movement of the sheet objectives with that movement to create three-dimensional XYZ mosaics of our large tissues. So in order to create a nice thin sheet, this system employs a tag lens, which is tunable acoustic gradient. It allows us the light sheet, instead of just using the waste with a normal Gaussian beam, this gets scanned very rapidly through a large field of view, the full field of view of the objective, which allows us to get a nice thin sheet with a moderate to high NA that gives us thinner and better axial resolution. The mags available on the system are between 2.2x and 8.9x, and we can do refractive indices from 1.33 to 1.58. Notice sample holders will give us samples from, say, a whole mouse brain in the 22 by 22 millimeter cuvette. We have a mid-size cuvette for things such as rat brains, which is 30 by 30 by 60 millimeters. And then we have what we call the mouse coffin, which will give us 30 by 30 by 10 centimeters. And we have used this. It's fantastic for large stuff. Importantly, the dry objectives. So we're using dry objectives for sheets and for the detection because we have high refractive index. We do calibrations on this to accommodate for the changing focal distance through the at depth at high refractive index, which allows us to maintain focus on the sheet as the samples move through that sheet. So I'm going to give a couple of quick examples, and I'm going to show some actually really old school stuff and modern version of that old school on model organisms. I'm going to show